So again, welcome and thank you for joining the second edition of the FARA Flash Talks in honor of the FA Awareness Month, the month of May. These short format presentations are sort of celebration to the young investigators with their research and engagement in the research for Friedreich ataxia towards a better understanding of the Friedreich ataxia and towards better development of future therapeutics. The format of the flash talks is made in a way that anyone without scientific background will be able to understand, and we really hope for an engaging edition, like the previous one 2 weeks ago. For the ones who haven't had the chance to watch live the flash talk series number 1 2 weeks ago, it has been recorded and posted on the YouTube channel of the FARA. I really encourage you to go and watch it, and today's edition will be recorded and posted later on as well. So as I said, today's session continues that tradition to highlight the passion of the early career investigators, focusing on the young investigators who are FARA funded across the globe. We have researchers from the USA and from the UK today, and we are delighted to have two moderators today who will present the speakers, their topics, and start engaging the questions. And anyone connected is invited to raise questions in the Q&A box as well. Our moderators today are Niamh and Liam. Niamh is from Ireland and Liam is from the USA. Both have FA. Niamh is a journalist. She has been writing many papers and advocating on rare diseases and on living with disability. I've had a chance to meet Niamh in person in the context of an activity in the European Commission, actually, raising awareness on rare disease and disability. Niamh, thank you for joining. And Liam is based in New Jersey, working in New York in actuarial science. And he shares something with one of the presenters today. He's a big athlete, and he has made it recently as a first FAer achieving the triathlon and an aqua bike. Again, Liam, thanks for joining. And from here, I will pass it first to Liam, who will introduce Anna and take it from there. Thank you.
Thanks for having me. This is great. So, the first speaker is going to be Anna Stepanova. Her research program is going to be she's a research associate in neuroscience of Weill Cornell medicine. The title of her presentation today is going to be A Tale of Two Cities: Navigating the Map of FA Cardiofibrosis. In this talk, Anna is going to explain a little bit about her collaborative project between Dr. Manfred and of Cornell and Dr. Payne of Indiana University. And this research program is going to help us to identify the early triggers of FA heart injury. So please, Anna, let us know about your program.
Thank you, Liam. And I believe in the beginning I have to say what brought me to science and what brought me to FA research specifically. So I'll give a brief introduction. I think I always loved math, data analysis, and by the time I was in college, I became interested in mitochondria, and this is what brought me from Russia to the United States as a visiting researcher during my PhD program. I was a visiting researcher at Giovanni Manfredi's lab, and I knew nothing about Friedreich's ataxia at the time I joined this lab. But then in 2015, I watched the documentary, The Ataxia by Kyle Bryant. And in 2016, I participated in my first Ride Ataxia bike ride. And since then, I tried not to miss a single ride. And when I participated in my very first bike ride, it was my first 25-mile ride. I met a lot of people, both researchers, doctors, as well as patients and their families. And on my bicycle, I started asking myself a question: how can I contribute professionally to the field? It took me some time to figure it out, and 5 years ago, I became an FA researcher, and that FA project became my main project in Giovanni's Manfredi lab. And then FARA supported our research through postdoctoral fellowship, and we started studying fibroblasts and fibrosis a little bit, and the project that I will present to you today is the continuation on the work that we've been building for 5 years in the lab so far. Should I begin with my presentation?
Yeah, you can go ahead and put up your slide.
I hope you can see the slide. I want you to close your eyes and think about your heart. How does it feel? What is it doing right now, beat by beat? How it expands and fills with the blood, and then how it contracts and pushes the blood to your body. I think we don't pay attention to our hearts most of the time until some problems arise. And many people on this call know that in Friedreich's ataxia, heart problems begin quite early, since the onset of disease. And one of many features of cardiac complications in Friedreich's ataxia is cardiac fibrosis. However, until recently, we didn't have tools to study this problem at the level of cells, at the level of communication between the cells, and we didn't understand what drives it, how we can prevent it, or maybe, if possible, how we can reverse some of the changes that happen at the organ level. But before we dive into the problem, I want you to imagine your heart as a city. In the heart, you have different cell types, and they perform different functions. So I want you to imagine a city with a few distinct neighborhoods. Your hard-working heart muscle cells, or cardiomyocytes, are the ones that serve as a pump. They expand, they contract, and they keep this blood flowing through your body. However, cardiomyocytes, or heart muscle cells, cannot do their work alone. They have some supporting cells. They have immune cells that would form, let's call it, medical district. Those cells will respond to any crisis that happens in the heart, and then they can send the signals to the neighbors so the neighbors can respond accordingly. You also have, let's call it, maintenance quarter. This will be fibroblasts. It's the same cells as you have in your skin. And sometimes when you get a scratch, in a few days, you see how the scratch starts healing and the scar tissue forms. A similar process can happen in your heart. And the cardiac fibrosis is the formation of this scar tissue in the heart. So instead of nice and elastic tissue, you can start having cardiac tissue a little bit more stiff, so it cannot function as well as in healthy conditions. So now let's build upon our city analogy. When you have a healthy city, all neighborhoods work in harmony. They have communication roads, they send the signals to their neighbors, they communicate that we are happy, we can work well together. However, in FA, you have a major energy crisis, and it becomes as if the city lost its power. And this muscle district starts sending signals to everyone around that something is wrong, we have to respond. And cells like fibroblasts from maintenance quarter, they go on the road and they start restoring the tissue. But sometimes they become overactive and they do a little bit too much of the road work, and some of the paths become closed and some of the new paths begin forming in your healthy heart tissue. And we want to understand, first of all, what signals the muscle district sends to its neighbors that triggers all these processes in cardiac fibrosis. How do these cells respond? Do the new cells form after they received communication that something is not right? And most importantly, how we can control or, if possible, reverse the formation of the scar tissue in the heart. In this collaborative study between Weill Cornell Medicine and Indiana University, we look at the mouse model of FA cardiomyopathy. It's a model in which frataxin is being removed only from cardiomyocytes. And this is enough for the heart to start experiencing problems. And even though at 6 weeks, the heart still looks healthy, by week 10, in those mice, they have massive cardiac fibrosis, and then later on, they die from complications. So we try to understand how the cells look like at 6 weeks when everything seems healthy and happy on the surface, but some processes already are in place and they will drive the changes that we'll see by week 10. And right now we are building those maps. You can imagine that we are building this satellite-like maps of the hearts. And then we will look into communication between different neighborhoods on these maps. We will try to understand how these cells respond to the development of cardiac fibrosis. And if possible, we would try to identify any potential ways to interfere with this process. Because that's not all. You have to understand that this technology has been developing for the past around 10 years. And by now, there are already existing maps for some other heart problems, both in mice and in humans. And we can borrow the knowledge that researchers already gained from those models, and we can try and apply either the same therapeutical intervention, or if we find that the FA is truly a special in the heart disease, then we can try to develop therapies that are selective to FA. And I hope that people who participate in ride Ataxia rides either as riders or as families will continue to do this, because this work wouldn't have been possible without that event. And I also would like to thank all my collaborators, both from Weill Cornell and the Indiana University, as well as my Toastmasters clubs. Those are clubs for public speaking that helped me to polish this talk.
Thanks so much, Anna. And Anna, how exactly, because now it's flashes to my mind, how exactly do you plan for this program to move forward toward a cure or a treatment?
So right now we are still learning what exactly is happening in the mouse heart when cardiac fibrosis develops. First, we want to understand how these cells communicate with each other and what exactly happens when they say that something needs to be done. When we identify these pathways, we will see if we can have any pharmacological intervention to either stop it from happening, or maybe if something is reversible, how we can treat it so that we can get back to a healthy condition. But right now, we are still in the very preliminary stages. We need to understand what is happening first before we can develop any avenues for the future intervention. And we hope that our study will advance our understanding of FA cardiac fibrosis.
Okay. And where we're going from looking at your presentation, I mean, that you just gave, you're using a mouse model where heart disease develops very quickly, and then your study would then take two specific time points, one before the scoring and then one after. So why exactly this model for the time points?
That's an excellent question, Liam. We chose this model because it has a lot of features that are observed in FA patients. If we look at the heart, so there is a formation of the scar tissue and some other properties of the heart also recapitulate what happens in the patients. That was the primary choice for this model. We also know how exactly the problems in these mice develop on a timeline. We wanted to catch right before we see any visible outcome, and then at a very late stage when we already see developed cardiac fibrosis. When we look at the earlier time point, we can see what programs are already in place that will trigger all this unfortunate sequence of events that will lead to the development of fibrosis. And these early time points will allow us to look at any potential pathways to stop cardiac fibrosis from happening. But on the other hand, when we look at the later time point, we will see what exactly is the destination and what new cell types appear, what kind of fibroblasts become abundant in the heart tissue, and maybe we will be able to identify some populations that we can treat so that they become less active. So we wanted to understand both the progression and also some points either to stop cardiac fibrosis or maybe to try to identify some possible target to reverse this phenotype if possible, because it's still unknown at what degree we can control the development of fibrosis if we can go back to a healthy state.
That's incredibly important, and that would be amazing if we were able to control the progress of the fibrosis. And then also, I mean, for the FA heart specifically, we have a lot of issues going on there. Why exactly the focus on fibrosis? Is it that it's like the most easy to track the progression of, or is it the most easy to treat it or delay the progression of fibrosis?
Thanks, Liam. In our case, we came to cardiac fibrosis project because we already started finding some interesting things in patient-derived fibroblasts. So we've studied those cells extensively, and we thought that it would be a natural continuation of our previous study, but in this case, we would focus on the tissue which is relevant for the disease, because skin fibroblasts are not something that is really affected in the disease itself, but it was an excellent cell model that gave us some insights into the directions that could be much more relevant to the FA community and then to the cardiac problems.
That sounds perfect. I can't wait to hear more from your program and from all that you find at the end of your project here. I'll give it back to the panel now.
Well, thanks, Leah. Thanks, Anna. Anna, we have two notes in the chat. First, from Mary, just congratulations, loving hearing about your first ride ataxia experience, how powerful, and thanking you for letting your riding experience inspire your research journey. And we have a question from Sanjay Bidichandani asking if you're able to identify single cells with 0, 50, and 100% frataxin levels immediately after induction of knockout, or the knockout happens fast and drops quickly from 100 to 0.
I hope that by the time for the ICAR conference, I will be able to answer Sanjay's question when hopefully I will see him in person. And thanks, Mary, for encouraging words, and also looking forward to your presentation.
Thank you. We move to Niamh and Nick.
Yeah, thank you, Miriam, and thank you, Niamh and Anna. So I'm presenting Nick Stachowski, postdoc at Children's Hospital of Philadelphia, and the title of his project is Investigating Sensory Motor Pathology Using Two Mouse Models of FA. But before we get into that, Nick, can I ask you what first got you into FA research?
Yes, of course. I'd be happy to answer. Generally, it was an interest in science that I've taken through all of my education, and eventually, getting into the real world post-college, I had worked like normal jobs, big pharma, but kind of always came back to academia. So, grad school, going through spinal expertise and such, the translation over to FA has been really nice. So there's a lot of crosstalk between the fields, and I'm trying to leverage this to help.
Great. Okay, so feel free to get into your project whenever you feel.
Yes, thank you. Okay, hopefully you can see everything fine. And yeah, as I was saying before, I had gotten into FA. Mostly I needed a job. I had a very specific set of skills coming out of the PhD. I wanted to keep working on ideas, and ideally my ideas. And I found a lab here with Eric Marsh, Don Joseph, and David Lynch, where they're kind of letting me explore these ideas. And so far, it seems like FARA thinks they are good ideas, but we can discuss that later. So I'd just like to say that I had many options of where to go, and my first COE meeting for FA here at CHOP, I saw how good of a community this was, and it was very inspiring to keep working in this field. So just so you know, and it's a really nice community, and thank you for having me. So I'll get into my talk, and there's not much background. We're all pretty familiar with FA, so I'll kind of just be jumping into my project and how it was built up. And this starts with a bit of history. So this dates some centuries ago, back to the initial characterization of Friedreich's ataxia. But suffice it to say that we now know that there's significant atrophy that occurs in the spinal cord. So there's loss of these dorsal columns, and this is where a lot of these tract fibers are projecting to the brain, and then some coming back down to the spinal cord as well. Fast forward some centuries, and we can see with some functional imaging studies that the spinal cord is indeed shrinking, so significant pathology happening down here. But when we go to look at the brain and circuits that control movement in the cerebellum, there's relatively little cell death occurring, and it seems to be very special or focused on the dentate nucleus of the cerebellum. And without getting into what each of these structures are doing, we can already see that there's differential vulnerability to FA and the loss of frataxin. And this is very tricky because we know frataxin's kind of everywhere. So for the context of my talk, I'm really worried only about frataxin in neurons and comparing these between models and different phenotypes. So I said frataxin's everywhere. We know there's a wide range of symptoms, but almost always there's sensory neuropathy and motor deficits. So this is where I start looking to the spinal cord, because both sensory reactions and all of our movement is really going to be relying on the work of spinal interneurons. Even if the cerebellum is healthy, it still requires an intact spinal cord. So my focus has really been on these excitatory and inhibitory spinal cord interneurons. Let me just get a laser pointer. Oop. Never mind. Sorry. Can you still see this? Or
We can
okay.
see the slide.
All right, I'll not click anymore. So it's not like the brain, though. The spinal cord is really a heterogeneous mix. So you can see different colors of neurons here. These are each different types of excitatory neurons and different types of inhibitory neurons. There's been a lot of work recently to take out each of these neurons. The name doesn't matter. It's just a way for us to go back and see who is doing what. So by taking out selective populations of excitatory and inhibitory, we can then look at a mouse and see how it's disrupted its motor functions. And kind of by reverse engineering this, we have a good idea of what each of these types of neurons do in the healthy condition. So with this in mind, I came to CHOP and started working on FA and kind of got the sense that we know that the spinal cord at late stages of the severe disease is severely affected, but this progression and how it got that way is not so well known. And then there's some recent work from various models showing that if we give frataxin back to certain tissue, that the nervous system can restore itself. So this is just highlighting one study where you take frataxin out of the proprioceptive fibers, which is shown, I wish I had my laser pointer, but on the right, these blue fibers are coming down, initiating and helping keep movement going. So these are lost early in the disease. You can restore frataxin and rescue some of these motor deficits. So my next natural question was, what about central circuits? If we targeted these pathways centrally, are they still there? And can we treat the disease before it's progressed? So now I'm going to introduce my two models. The names are kind of long, but we have the silencing or knockdown of frataxin in the DOX animals, and then the Kiko, which is a knock-in, knockout. And rather than having names, I'll just describe them. So on the left, it's a very aggressive phenotype. The mice are otherwise healthy until 4 months of age, and then we start removing frataxin very aggressively. They'll develop phenotypes rather quickly, and they end up losing almost all of their frataxin. On the right is the Kiko mouse. It's a more modest phenotype. They have disrupted frataxin expression from birth, and then later on into life, they'll start to display symptoms kind of variably. So with both of these models, I chose 4, 8, and 12 weeks for the aggressive phenotype, and 4, 8, and 12 weeks are months for the more mild phenotype. And so each of these mice are subjected to a battery of behavioral tests, just so I get an idea, are they pre-symptomatic, post-symptomatic, or asymptomatic? And these behaviors are not all, but kind of give me a composite score to show that these mice are starting to show symptoms of FA. So what do I do with the mice? Once I have the behavior, I kind of put it in my pocket, keep it for later, and then at the end of their experiment, I'll take the mouse and go one of two routes. So focusing on this left one first is electrophysiology. So I'm going to record electrical signals from the neurons that I had shown previously. And this involves preparing solutions which mimic the brain's fluids or artificial cerebrospinal fluid. You make very thin sections of the tissue and then record from the cell using a glass pipette, which is shown here in green. and then the cross-section of the spinal cord with red neurons shown, which I'm targeting. So you can see they're very small. The mouse is very small, but this is very valuable data. And really what I'm hoping to do with this is to get measures of cellular excitability, plot them against the severity of motor deficits. And I'm hoping to be able to show that cells with higher excitability belong to the mice that are performing the best, and vice versa, where for losing levels of excitability, we'll probably be showing more symptoms of FA. So this is just for the 8 months, but of course I'm doing for 12 and 4 months. These aren't real data points. It's just showing what I'm hoping to see. And then for a separate set of animals, it's histology, where I'm taking the spinal cord in a similar fashion, slicing it, but now probing for molecular markers to get an idea of the identity of these neurons. So to complement how they are physiologically performing in the body, I can then see what do these neurons look like under the microscope. And so this is just showing you a bit of what it looks like, where you get these very thin, like pepperoni slices of the spinal cord or brain. You can stain for various markers. Here I'm just showing red and green, where we have neurons in red, and then afferent sensory fibers in green. And again, what I'm trying to do is compare changes in the number of neurons or fibers in the spinal cord. How does this relate with behavior, and how does it relate with the electrical recordings from the other arm of the study? And this is actual data from just one cohort of from a recent cohort, where we have an 8-month-old group of control, KI wild type, and then Kiko FA mice. And this is very early days, but these mice are not really showing symptoms, but I'm already seeing decreases in the number of neurons without a change in the number of fibers. So just to summarize that, I'm seeing differences centrally before I'm seeing symptoms behaviorally. It'll take more data till I can be more confident, but it's early days. And then just to wrap things up, I want to be able to put FXN back into the neurons that need it and the ones that are lost first. So just again, the schematic diagram, you can pick your favorite one. These are just inhibitory, operating off the hypothesis that the loss of the inhibitory populations occurs first. And then on the right is just showing some staining data where I can stain all inhibitory neurons with the marker PAX2, and then more selectively stain a smaller population of these inhibitory neurons, these ones being the ROR betas. But just for proof of principle that of all these neurons, we can really fine tune and pick out the ones we want to manipulate. And my grand idea with this is that I can use these mouse models to correlate both physiology, histology with behavior. This will give me a cellular, circuit, and behavioral level of analysis. And then finally, hopefully by doing this work, I can give more insight into the mechanism of how this disease is progressing, and can we prevent it from progressing further, but really ideally, can we revert it back to being functional again? And so with that, I thank everyone at CHOP, my lab, and thank you all for listening. And I'd be happy to take any questions.
Thank you, Nick. That was a fantastic presentation.
Thank you.
I just have a few questions here, and then yeah, we can look through the chat, or if any panelists have questions, we'll get to them. But so my first question is, why did you pick these particular mouse models?
Yeah, sure. It's a great question, and there seems to be more mouse models every day or every year at least. So to be honest, we have these mice going in the animal facility, so I started working with them. But the better answer is that I think it most accurately represents a human condition in separate ways. So the Kiko and the dox kind of complement each other. I like to say that the Kiko is this more developmental view of it, with the dox being more degenerative. So I can kind of answer two questions at once. And yeah, I could say a lot more, but yes, that's the reason why.
And do you expect to see central spinal changes before or after changes to peripheral sensory?
That is a hard question. It's a very interesting question, and it's kind of, I have a lot hinging on it, but I think the better answer is both. Like, we know that the sensory fibers are degenerating, but we don't know what that loss does to the spinal interneurons, and it could be a case that we lose central neurons and things start accelerating. So I'm trying to tease this apart currently. Thank you for the question.
And then my final question is kind of a more broad one, I suppose, but where do you hope this research will lead you in the future?
I think, with gene therapy, AAV, these types of things, like if we're trying to target the cerebellum, I think the spinal cord's a better target. So I'm trying to make that pitch, maybe revolutionize treatment options. And ultimately, there are cases where sometimes you have motor deficits and you don't have the other symptoms. So this could really open up some very specific treatment options for people. And I just want to help. So that's it.
Sorry, I
Okay.
muted myself there, but fantastic. Thank you
Yeah,
so much.
thank you.
Miriam, I'll pass back to you.
Yes, thank you. And again, Nick, to you as well. We have thanking notes from Mary as usual. You can read it in the chat. Dr. Lynch is her neurologist since she was diagnosed about 25 years ago, and your research and dedication to biology and science really gives a lot of joy and hope. And I'm sure not only to Mary, but to all the members of the community who joined today. And we have another thank note from Ari and Reiki as well.
Thank you very much.
Okay. Niamh and Marta, I'll pass it to both of you.
Yeah, and our third speaker of the day is going to be Marta, who is a postdoc at the University of Oxford. She's going to be giving us a presentation called How Friedreich's Ataxia Affects the Heart and What We're Doing to Help It. Before we start, though, I would like to hear from Marta. Why exactly did you get interested in Friedreich's ataxia research?
Thank you, Niamh, for the kind introduction and for being the moderator for this talk. Regarding your question, I will start from the beginning. I will say that I have always been a very curious person and I'm very interested in understanding the process that happens inside our bodies. That curiosity is what led me to study biotechnology. I have always been passionate about research, creating new knowledge, and trying to understand what happens in disease. Because I truly believe that the better we understand diseases, the better we can treat them and hopefully develop cures in the future. And I think creating new knowledge is the only way that we can move forward. After my biotechnology degree, I completed a master's degree in molecular biology and biotechnology, always continuing along the same research path. And after my master, I started my PhD, where I worked with human stem cells. There are very special cells because they can be converted into many different cell types depending on what you want to study. And they are an incredibly useful tool for researchers like me. And I worked with these models for around 6 years and became very interested in using them to study human disease. After that, I decided to move from Spain, where I'm from and where I completed all my studies, to Oxford in the United Kingdom to start my postdoctoral research in Friedreich's ataxia. And for that, I joined the Topfer group about 2.5 years ago. And since then, my work has focused on understanding what happens in the heart in Friedreich's ataxia and using these human stem cell models. it is what my presentation today will be about. Yeah, let me share my screen.
My goodness, by the way, that is quite the resume, Marta.
Thank you. Could you please confirm that you can see my presentation properly? Right.
Yeah,
Thank
you got it up.
Before I start, I would also like to thank FARA for giving me the opportunity to present today in this FARA Floss talk in the Awareness Month of Friedreich's Ataxia and share my research with patients and families. I think this is a fantastic initiative because it helps connect researchers with Friedreich's Ataxia community, and I'm very happy to be part of it. To put you in context, I'm based at the University of Oxford, as I said before, and my role as a postdoctoral researcher in the Topfer lab is to better understand how Friedreich's ataxia affects the heart. The Topfer lab focuses on cardiac physiology and disease modeling, and it's a large group that study different heart disease, and I joined the group to lead the Friedreich's ataxia research project. To help explain Friedreich's ataxia, I like to use a simple analogy. In a healthy person, the frataxin gene here represented as one page of a manual of instructions, where the whole book represents all the genetic information in our cells that these genes works normally. We all have two copies on these instructions, one inherited from our mom and one from our dad. In a healthy person, both copies of the frataxin gene are written correctly and work properly. These instructions manual produce workers inside the cells. In this analogy, the workers represent frataxin, the protein that is reduced in Friedreich's ataxia, and helps cells produce energy. When the manual works properly, the cells can produce enough workers, and these workers help the cells to generate enough energy to function normally. All the cells in our body need energy to work properly. However, organs like the brain, the heart, and the muscles are specifically demanding because they need a huge amount of energy all the time. In my case, I will be focused on the heart. In a healthy situation, heart cells have enough workers and enough energy so they can function properly. On the other hand, in Friedreich's ataxia, both copies of the instruction manuals are altered or damaged. Better. The page of the instruction manual is not exactly the same that in the healthy person. Because of this, the cells cannot produce enough workers here. And as a consequence, the cells cannot produce enough energy and they struggle to do their job properly. This is especially important in the heart because it's one of the organs with the highest energy demand in the body. And over the time, it becomes severely affected by this lack of energy. Heart cells need, as I said before, a huge amount of energy to keep beating through our lives. To help explain this, I would like to compare our cells to power plants. In a healthy situation, a power plant has enough workers, the machines run properly, and enough energy is produced and distributed for the cells. This is especially important for heart cells because they are highly energy demanding, as I said before. But in Friedreich's ataxia, there are too few workers. And this, as I explained before, and the machines don't work well and don't work efficiently, and not enough energy is produced in the power plant. Over time, this lack of energy can damage cells, especially heart cells, and contribute to the heart problems we see in Friedreich's ataxia. Over time, this lack of energy can affect the heart very severely. Around 60% of people with Friedreich's ataxia develop heart disease, and the most common problem is something called hypertrophic cardiomyopathy. To understand this, imagine that the heart is like a balloon with 4 chambers, and one of these chambers is responsible for pumping blood from the heart to the rest of the body. In Friedreich's ataxia, this chamber can gradually become thicker and enlarge over time. In the walls of these chambers, the heart muscle cells grow bigger, as we can see in this video, and in some people, scar tissue can develop. And is represented here as the white things. Even though the heart muscle becomes thicker, the overall size of the heart often stays similar. This means that the chamber cannot hold as much blood as before, make it harder for the heart to pump blood through the body efficiently. This is a comparison between a normal heart and a Friedreich's ataxia or hypertrophic cardiomyopathy heart. The heart can also become stiffer, meaning it cannot relax properly between contractions. However, we still do not fully understand all the mechanisms behind this process. Here you can see a summary comparing a healthy heart and a Friedreich's ataxia heart. As we just saw in the animation, over the time, the heart muscles can become thicker and stiffer in Friedreich's ataxia, and this yellow arrow shows. In a healthy heart, the muscle has normal thickness, is flexible, and it can pump blood efficiently around the body. In Friedreich's ataxia, the heart muscle can gradually become thicker, like you can see here, especially in the walls in the main pumping chamber of the heart that I have shown before. The heart can also become stiffer, meaning it cannot relax properly between contractions. And in some people, scar tissue, also called fibrosis, that Anna explained before, can build up over time, represented here with these white structures. We know all this information, but we still do not fully understand all the mechanisms behind these processes. And that is why research is very important. In my research, I wanted to create cells that resemble the heart cells. And to study this, I generate human heart cells in the lab to better understand what is happening inside these cells. To do this, I use stem cells that they have been generating from adult cells donated by healthy individuals and Friedreich's ataxia patients. These stem cells are very special because they can be converted into many different cell types in the body, including heart cells. I use these stem cells from both a healthy donor and from a patient, and then I differentiate them into heart cells. And I call it like lab-grown cells. And importantly, they also keep the properties and genetic information from the donors, including all the instruction manuals we discussed before. Using this approach, we can generate healthy Friedreich's ataxia heart cells in the lab and compare them side by side with Friedreich's ataxia 1. This allows me to study what changes in Friedreich's ataxia heart cells and better understand why the heart becomes diseased. Using this approach, we can compare healthy and Friedreich's ataxia heart cells side by side and study key features of how heart cells function. We can produce a large number of these cells in the lab and study many different aspects of disease over time. For example, we can study how strongly the cells contract and relax, how they communicate using electrical signals that allows heart cells to beat properly, how contraction is coordinated, and whether this process is well regulated or impaired, and which genes are turned on or off because the frataxin instructions page is altered. This platform, developed in the Töpfer lab, also allows us to test potential treatments directly in Friedreich's ataxia heart cells and see whether we can improve their function and make them behave more similarly to healthy heart cells. All of this helps us better understand what changes in the disease. And the better we understand these mechanisms, the better chance we have to developing more targeted therapies in the future. I would like to finish my talk saying thank you very much for listening. Thank you to the audience, and thank you to FARA for supporting and funding this project and for organizing initiatives like this, FARA Flash Talks, to raise awareness about the importance and need for research. And I will now very happy to take any question that you might have.
Thank you so much, Marta. Is there anything specific about pluripotent stem cells that are going to make the study of FA and the FA heart a little bit easier?
Yeah, it's a good question. Yeah, stem cells that are generating from adult cells, such as skin or blood cells, they can be reprogrammed from being adult cells back to into stem cell stage. And at this stage, the cells become pluripotent, which means that they have the ability to be converted into almost any cell type in the body, including heart cells. And this is very useful because Friedreich's ataxia samples from heart is quite difficult. And also, the samples are quite limited, and you can't grow them into the lab for a long period of time. So these stem cells allows us to create patient-specific heart cells in the lab, and it's like to have a Friedreich's ataxia heart in the dish. And we can study in more detail and compare healthy and Friedreich's ataxia cells side by side and test potential treatments as well directly and see if these Friedreich's ataxia cells improve their function.
That sounds really good. That sounds really promising, these pluripotent stem cell models.
Thank
And
you
then
very much.
I mean, you did touch on it at the end, but is there any specific goal or any hope at the end of all of this research for like a pushing for like do you have anything specific that you plan to get done, like maybe you mentioned like a therapeutic to help it, or like is there any treatment that you are eyeing at the end of this, or is it mainly just the actual understanding, because we really don't have any?
You're right. Yeah. There is no treatment. There is no cure at the moment. I know that I'm working in our lab also. We are working to find a cure and to find new treatments. It's not that easy, but we are working on that, and hopefully in a few years we can find the cure.
And this is more of a statement, but I mean, for the panel and anybody watching, like, I mean, I've spoken to countless other cardiologists that are specifically looking into FA. A lot of them don't even agree that FA cardiomyopathy is entirely hypertrophic. So this sector really needs to be looked at, and I'm really happy that Marta is taking a look into this with her fantastic models. Thank you very much, and I'll return it back to the panel.
Thank you, Liam.
Thank you, Liam. We have a question in the chat. Do you want to take it, or I take it, Marta? The one from Sanjay, if you can see it.
Oh, yeah. Is there a plan to co-culture with cardiac fibroblasts that are themselves frataxin deficient? Yeah, that's the plan also. Not only work with heart cells that are cardiomyocytes, the plan is also to work in 3D models with different cell types, not only fibroblasts, but also endothelial cells and other cells that are involved in the heart, because heart is made by different cell types. That, is the next step that we have in mind and we are working on at the moment.
Great, thank you, Marta. And we have one more time from Mary, a thanking note with gratitude to you. And she really loved your graphics that you used in the presentation with the analogy of the workers and the energy, very useful. And she's really grateful to your commitment. Thank you.
My pleasure. Thank you very much.
Any other questions from Niamh, Liam, or the other researchers to each other? Well, I don't see other questions in the chat. With that, I would like to thank you on behalf of FARA. Thank you for the speakers for sharing your research. We felt your commitment and passion. And thank you for the moderators who really led the session. Manasa and I didn't do a lot, so really thank you. And of course, thank you for all the persons living with Friedreich's ataxia, for the advocates, the families, also the clinicians and the researchers. The session today is another example about how important is sharing and collaborating. And without all this collaboration, we wouldn't have been where we are today in the progress and research in Friedreich ataxia. So again, on behalf of FARA, thank you. And let's connect soon on another opportunity with FARA webinars and hopefully next year for FA Awareness Month. Thank you.